تالتة ثانوي هانت خلاص، أنا قد التحدي، خطوة كمان والبكرة هعدي. تالتة ثانوي، تالتة ثانوي.
Welcome dear student to our channel مدرستنا 3 under the supervision of Ministry of Education. أعزائي طلاب الصف الثالث الثانوي، أهلا بكم في الحلقة الثانية من المراجعة النهائية لمادة الأحياء باللغة الإنجليزية. Okay. Dear student, this is the second episode that's related to final revisions about the immunity and DNA and RNA related to molecular biology. Please pay attention, please, and focus me in this. Why? Because the very important revisions that help you to solving the problem that faces you in the exam MCQ and another type of questions this year. It's called essay. That's right. Okay. Immunity living organism. What is the means of defense to which the plant resorts when it's found in resisting a microbe that's spread in some of its tissue? Hypersensitive response when the plant infected with the microbe and pathogens, and this microbe and pathogen invasion the plant. The plant must be get rid of this infections or pathogens. Why? To avoid the infections in the healthy cells. So the plant tend to specific types of the immunity. It's called hypersensitive response. So the best answer is hypersensitive response to get rid of damaged tissue, to avoid infections, another the tissue is infected by the invasion of the microbe and pathogens. B, formation of cork. No, it is the specific type of the immunity. It's called structural immunity. Pre-existing structural defense, induced structural defense, and antimicrobial chemical, what's called biochemical immunity. The immunity in the plant is divided to, number one, is structural immunity. Pre-existing structural defense. For example, cellulose, its depositions, it contains the cellulose, its depositions in the cell walls of the cell play an important role in the pre-existing structural defense. Okay? Hairs, that's spiny, it's found in the epidermis cells to prevent accumulating in the waters. Okay? Pre-existing structural defense. Induce the structural defense. Number one, formation of the cork layers as a result of invasion of the microbe and in the plant is cutting or tearing. Okay? Formation of the tylosis, overgrowth protoplasm that adjacent the protoplasm between the xylem vessel and trachea. This obstructs the invasion of the microbe and pathogens. It's called formation of the tylosis. Obstruct the invasions. This is the specific types of the infections, the formation of the tylosis after infections. After the infection, the plant forming the tylosis. That means that the microbe invasion the plant. The microbe invasion the plant. It's called induced structural defense. Formations, the gums. Hypersensitive response. Okay, cellular immunity structure. Some morphological change is occur in the cells as the result of invasion of the microbe and pathogens. For example, the cell wall is surrounded by mycelium of fungus to prevent spread inside the plant. Again, returning to these questions, what is the mean of defense to which the plant resorts when it's found in resisting a microbe that's spread in some of its tissue? Hypersensitive response is the best answers to get rid of the damaged tissue to prevent spreading. Formation of cork, induced structural defense. Gum depositions, induced structural defense. Formation of insulators covers, it's called specific types, it's called the cellular immunity structure. Cellular immunity structures. The best answer is A. That's right. Another question. The formation of tylosis is similar to the deposition of the gum, the N. The shape and the structures, no. Tylosis, overgrowth, protoplast, that's adjacent between parenchyma cell. protrude from xylem vessel to trachea. Between the xylem vessel, this represents the xylems, vessels, and the trachea. Both of them, between them, it's called tylose. It's formed to obstruct the invasion of the microbe and the pathogen, and obstruct the waters. So it's different in the shape and the structures. The site of formations, no difference. The formations after the infections, well, number D, prevent the entry of the microbes. Both of them, the microbe enter already. But the formation after the infections, number C, it is the best answer is C. It's formations after the infections. Okay, that's right. Another questions. What are the chemical substances that may be used to inhibit the action of hepatitis C virus in the liver tissue? Immunity. There is specific types of the assisting chemical substance. Number one, chemokines. They are important recruits or towards the macrophage, the site of the infections, and they prevent reproduction of the microbe and pathogens. It's called the chemokine. Another assisting chemical substance is called complements. A specific type of the proteins. Okay? Eliminate the microbe and pathogen after conjugating with the antibody. When it's conjugating with the antibody, lyses the antigen and the microbe and the pathogens, and introduce to the phagocytic cells as macrophages, basophilus, neutrophilus, to engulfment. The complements. Interleukin, a specific type assisting chemical substance. Mediate communication between immune system and cells, mediate communications between immune cells. Finally, interferons. It is the assisting chemical substance. It's infected, especially from the cells that infected with virus, to prevent reproduction of the virus and struggles its invasion of the microbe and the pathogen. So, the best answer related to these questions, what are the chemical substances that may be used to inhibit the action of hepatitis C virus in the liver tissue? Chemokines? No. Interleukins? No. Complements? No. The best answer, interferons. Why? Chemokine recruits the site of the infections, any infections. Interleukins mediate communications, activations. Interleukin activation helper T cells and the B cells, the production of antibody, not specific, the cell infected with virus. Complements lyses the antigen in microbe, not virus. But interferons specifically is secreting from infected cell with virus, so the best answer is D. That's right. Okay. Now we are going to focus about this lesson through the part. What are the chemical substances that may be used to inhibit the action of the hepatitis C virus in the liver tissue? Chemokines, interleukins, complements, interferons. The best answer is the interferons. Why? The interferons play an important role, inhibit the action of virus and secreting, especially from the infected cell with virus. Another questions. Please pay attention to me and focus on these questions through the part. That's right. Okay. To treat a person who was bitten by a poisonous snake, okay, by which of the following substance is better for injecting him? Okay, that's right. Again, to treat the person who was bitten by a poisonous snake. Poisonous snake is secreting a specific type of poisonous substance. By which of the following substance is better for injecting him? This person bitten by the snake. What is the best way to treatment this person? Interferons? No. Interferon related to the infected cell with virus. The poisonous substance that's secreting from the snakes, not related to interferons. Antibody is the best answer. Why? The antibody, when the person is bitten by the snake is secreting a poisonous substance reaching to the blood. Specific types, it's called antitoxins. By combinations between or fused antibody with the poisonous substance. The complements enter a group of the reactions and reduce the toxicity. After the poisonous substance that's secreting from the snake enter to the blood, the antibody combines with the toxic substance, and the complementers combine with the antibody, enter the group of reactions, reduces the toxicity. As that it occurs in the plant, it's called the detoxifying enzyme, antimicrobial proteins. This is at the same mechanism that's occurred in the human. It's called antitoxin. This is the mechanism, one of mechanisms that's done by the action of antibody. So the best answer is antibody. Interleukins, no. Chemokines, no. The best answer is antibody. That's right. Another question. What are the most abundant substances that are found in the tissue of an infected epidermis by acne? Acne, that's represent small pills that found in the face in human body in adult stage. This is the exposure to the many microbes. Okay, that's right. To avoid this, in this way, assisting chemical substance such as chemokines recruit the macrophages site of the infection. Why? These sites may be exposure to the infections through any microbes that enter through the pills. It's called acne. So this is the chemokines play an important role in the recruit the macrophages site of the infections to involvement by the phagocytosis. So the best answer is chemokines. Again, these questions, what are the most abundant substances that are found in the tissue of an infected epidermis by acne? Chemokines, it is the best answer. Why? To recruit the macrophages that site in the infections. Antibody? No. Antibody, the antibody have a many mechanism. Number one, neutralization. It's specific for the destroys the virus. Destroys the virus, okay, and prevent reproductions. Another, coagulations. A specific type of antibody, it's called IgM. It contains more than antigen binding sites, accumulating the antigens at the same antibody to facilitate its engulfment by the phagocytosis. Precipitations, it's occur between soluble antigen and antibody. It transfers insoluble antigen antibody, and it's precipitate to facilitate engulfed by the phagocytosis process. Another, it's called lysis. Lysis depending on the complements. When the antibody through the specific antigen binding site with the antigen. Specific type of complements binding with the antibody. Complementous binding with the antibody, and after the antibody binding with the antigen antibody with the specific type of antigen. Complementous lyses the surface of antigen and facilitate to the phagocytosis. When you see the complements and antibody, the facilitating play an important role facilitating phagocytosis, introducing this into macrophages, basophilus, eosinophilus, neutrophilus to engulfment by the phagocytosis process. Okay, that's right. Finally, antitoxins. As the last questions mentioned, the snake biting humans, so secreting a specific type of the toxic substance, antibody binding with the toxic substance, complementous enter the group of reactions, reduce this toxicity. So the best is answer in these questions. It's called the substances that are found in the tissue, infected epidermis by the acne, chemokine. It is the best answers, not antibody, not complementous, not interleukin. Another question. What is the role of the interleukins? Pay attention, please, in this question. Interleukin related to third line of defense. It's called acquired immunity. Acquired immunity related to third line of defense. If the first line and second line failure to resist the microbe and pathogens, the third line is called the acquired immunity through the immune response. What does it mean by of immune response? A group, a specific defense mechanism done by lymphocytes. Lymphocytes, B lymphocytes, T lymphocytes, NK natural killer cells. That's right. This is called lymphocytes. The acquired immunity is divided to specific types of the immunity. Humoral immunity related to productions of antibody. Cellular immunity depending on the receptor found in the membrane of T lymphocytes. Humoral immunity, it is the immune response through which P lymphocytes productions an antibody to resist against microbe and pathogens. To resist against microbe and pathogens. Okay, that's right. When the B lymphocytes binding with the antigen that found in surface in their membranes, B lymphocytes it contains MHC, major histocompatibility complex, found in it, binding with the antigen and presenting on its surface, following MHC antigen complex. At the same time, the macrophages engulf the microbe and pathogens. Inside the macrophages, a specific type is called MHC, major histocompatibility complex. The macrophages contain the lysosomes, breaking down the antigen to small fragmented and binding with the MHC and presenting its surface following MHC antigen complex. A specific type of T lymphocytes, it's called helper T cell. Through receptor found in the membranes, it's called CD4, cluster differentiation force. Binding both of MHC antigen complex in the surface of macrophages and the MHC antigen complex found in the P lymphocytes. It's activated through MHC antigen complex and secreting interleukins. This interleukin activates the P lymphocytes. The B lymphocytes is divided and differentiations a specific type of cells. Plasma B cells to productions antibody. Memory B cells remain in the blood approximately 20 to 30 years. And another types of the immunity is called cellular immunity structures. It is the immune response done by T lymphocytes through receptor found in their membranes to give a specific response against a specific antigen. The T lymphocytes, which is binding through receptor found in their membrane, CD4s, with the MHC antigen complex in the cellular immunity structures, it's secreting interleukin. Interleukin activations helper T cells. It's forming memory helper cells, and secreting a specific type is called cytokines. Cytokines activation both of humoral immunity and cellular immunity, and activation the cytotoxic T cells to production perforin and lymphatic toxins. It destroys the specific type of gene that's found in the infected cells. Returning to again and these questions, what is the role of interleukin? I explained now I am explaining this lesson. This is point related to interleukins. Interleukins activation both of helper T cells, TH, and B cells, both of humoral immunity and cellular immunity. So the best answer is activating helper T cells and B cells. The C is the best answer. What is the role of interleukins? Activating B cells only, activating helper and killer T cells, activating B cells and helper cells. The best answer is C. That's right. Okay. فاصل قصير إن شاء الله ونعاود لاستكمال حلقة المراجعة النهائية لمادة الأحياء باللغة الإنجليزية. انتظروني أعزائي الطلاب إن شاء الله بعد الفاصل.
تالتة ثانوي هانت خلاص، أنا قد التحدي، خطوة كمان والبكرة هعدي. تالتة ثانوي، تالتة ثانوي، تالتة ثانوي هانت خلاص، أنا قد التحدي، خطوة كمان والبكرة هعدي. تالتة ثانوي، تالتة ثانوي.
أعزائي طلاب الصف الثالث ثانوي، رجعنا لكم بعد الفاصل ونعاود لاستكمال حلقة المراجعة النهائية لمادة الأحياء باللغة الإنجليزية. Now we are going to focus on other questions that related to immunity in living organism. Follow me, please, and pay attention. That's right. Which of the following statement is correct about the natural killer cells, NK? Natural killer cells, a specific type of the lymphocytes that represent 20 to 30% from a white to the white blood cells. Okay? Natural killer cells found in the second line of defense and third line of defense. So it's not highly specific. Highly specific related to B lymphocytes, T lymphocytes. So the slow response. But in the first line defense, it's not specific. It's immune response, not immune response, but it is not specific. Okay? That's right. Which of the following statement is correct about the natural killer cells in NK? They are not specific for a certain type of antigens. That's right. They don't contain perforins. They are a type of T cells. Belonging to the third line of defense only? No. It's belonging to the third line and second line. It's wrong. Is there a type of T lymphocytes? No. T cells is a type of T cells. Helper T cells, TH. Cytotoxic T cells, TC. Suppressor T cells, TS. This is wrong. They don't contain perforins. That contain the perforin. It is the T cells secreting a perforin substance. It's perforated the antigen of microbe and pathogens. It's wrong. They are not specific for a certain type of antigen. The best answer is A. That's right. Okay. Another question. Label the opposite figures. This is a specific type of questions related to the another types of the questions in this year. It's called essay. Pay attention, please, and focus in these questions. This questions is very important to solving your problems. So, returns any questions that related to in this point. But MCQ, one choice only, A, B, C, or D. But in this particular type of questions, and focus in this point and to solving by writtens in your paper answers. Okay, that's right. Label the opposite figures. What are the heavy chain and the light chain and how they fit to each other? This specific type is called antibody. A specific type of protein, it's called immunoglobulin. So it's symbolized by Ig. What is the type of antibody? IgM, AGD. MAGD. Okay? MAGD. IgM, IgA. Okay. That's right. The antibody is called immunoglobulin, Ig. Consists of protein substance found in the plasma. It's called immunoglobulins, Y-shaped, as is found in these figures. Y-shaped. Two pair of a chain. Two long chains, it's called heavy chain. This is long chain, it's called heavy chain. And short chain, it's called light chain. Both of long chains and the heavy chains is connecting together by disulfide bond. The disulfide bond between long chains and heavy chain. Long chain and heavy chain. And between two long chain, disulfide bond. That's right. This region's number 4, it's called antigen binding sites. It's variables according to the type of antigen from antibody to another antibody by changing the specific type of antigen. Number 4. It's called variable regions, antigen binding site. This is called constant regions. Constant regions. Okay? Hinge regions. Open and closed. Hinge regions. Disulfide bond. Complement binding site. When the antibody binding with antigens. After the antibody binding with the antigens through a specific type of antigen. Each antibody has a specific type of antigens. The complement binding through the complement binding site. That's right. The antibody destroyed completely the microbe and the pathogen after conjugating the complements. Antibody bending through antigen bending site in variable regions, bending with a specific type of antigen. After bending with a specific type of antigen, the complement is bending with the antibody, lyses the surface of antigen and reducing into the phagocytosis process as macrophages, basophils, eosinophils, neutrophils to make the phagocytosis. That's right. Again, label the opposite figures. Number 1, variable regions. Number 2, constant regions. Number 3, disulfide bond. Number 4, antigen bending site. Number 5, light chain. Number 6, hinge joint. Okay? Number 7, complement bending site. Number 8, heavy chain or the long chain. Okay? That's right. What are the heavy chain and the light chains and how they bend to each other? Light chains, long chain. Short chain. Heavy chains, long chain. Both of them bending together by the disulfide bond. Both of light chain and heavy chain bending disulfide bond. Both two heavy chain or long chains bending by disulfide bond. How antibody differs from one another? By changing the variable region antigen bending site. When the antigen bending site is changing in the variable regions, a specific type of amino acid is changed. Why? Because the antibody is mainly composed of the amino acid. Enters the main structure and it's formed from proteins. That's right. What is meant by the constant and variable regions of the antibody? Constant regions, this is not constant to all type of antibody, not a change. Variable regions, it's that's found in the region number 1. Why? Because it is changing according to the antibody, bending with the antigens. So it's called variable regions. It's changeable. What is meant by the constant and variable regions for the antibody? How antigen-antibody complex is formed? When the antibody bending with the insoluble antigen is transferred into soluble antigens with the antibody, bending together, it changes insoluble antigen antibody. It's precipitate. When it precipitates, it has to facilitating engulfed by the phagocytosis through the macrophages, basophils, eosinophils, and neutrophils. That's right. Okay. Another questions? Molecular biology, DNA and RNA. Molecular biology, in this studies the structures, in this unit, structure DNA, function of DNA, replication DNA, genetic materials is called DNA, not proteins. As the first scientist in the past thought that the genetic material is protein, not DNA. Why? Because protein, it consists of 20 type of amino acid, but DNA, it consists only mainly of 4 nucleotide. It's called purine, adenine, and guanine, double ring. Pyrimidine, it's single ring, thymine, and cytosine. But in this experiment, that's proof that the DNA is a genetic materials. It's called bacterial transformations, bacteriophages, and the amount of DNA in living cells is constant, not variables. Constant in all at the same species. When it's fused, half number of chromosomes from male gametes with the half number of chromosomes from female gametes, producing the embryo, adult living organism. This is the amount of DNA is constant. Starting with the bacterial transformations. The scientist Griffiths proved that the specific type of 2 strains of bacteria is strain S. That means that smooth bacteria. Why it's named by this name is smooth? Because it contains specific type of genes responsible for formation of the capsules around from it to protect it from another immunity when invasion the humans, animals. This is genes forming the capsules to protect this strain of the bacteria, so it's called a moth bacteria, strain S. It's called virulent, has the ability to kill or destroy any living organism. Why? Because it's had the protections genes responsible for formation the moth capsules to protect it from the immune system. This strain is called virulent, strain S. Non-virulent, strain R, rough. That contains the gene that's responsible for the capsules to protect it from the immune system. This scientist bring mice, injecting the first mice with the strain S. Infected with pneumonia, a specific type of the microbe causes destroyed the respiratory system, especially to lungs. Infected with pneumonia and die. Strain S. Another mice injected with the strain R non-virulent, infected with pneumonia but not death. Struggle death. The immune system of all the mice struggle the strain R. Okay? Give the strain S and exposure to the heat and infected and injected with the mice. The mice not infected with pneumonia and still alive. Give this strains S that exposure to the heat or killed it by heat and injection another strain R to the mice. Some mice die. Why? Because the strain R's, it gains the genetic material of the strain S and the changing from non-virulent to virulent to have the ability to kill the mice. So the bacterial transmission is okay. That means that the genes that are responsible for appearance of the strains and that gained by the strain R non-virulent, it related to DNA. That means that DNA is a genetic materials, not proteins. Avery and his colleague tell us or told us this is DNA is contaminated with proteins. What they mean by the contaminated with proteins? It contains another amount of proteins, maybe histone protein. Maybe this is the reason for the transformations. So the scientists tend to crucial experiment, gave the strain S that exposure to the temperatures and exposure to the deoxyribonuclease enzymes, destroyed completely the strain S, okay? And infected, injected the mouse with the strain R, no bacterial transformations, it's okay. That means that the DNA is the genetic material. In bacteriophages, when the bacteriophage infected the bacteria, after 32 minutes, releasing 100 of the phages. This phages is labeled by the phosphors. This is the phosphors that found in the DNA. Sulfur found in the protein, found that abundantly 97% from the phosphor enter to the cells and less than 3% of the sulfur. That means that phosphor found in DNA, that means that the DNA is a genetic materials and the amount of D is constant in all living organisms. That's right. What happened when treating bacteria strain S, virulent, with the heat and the deoxyribonuclease enzymes, then transferring to the bacteria R? Bacteria R will die. No, not affecting the bacteria R. Bacteria S will acquire the characteristic of the bacteria R. No, bacteria R's is gains the genetic material of the bacteria strain S, not S gains. Bacteria R will be transformed into bacteria S. No, bacteria R will not be transferred into bacteria S. That's right. Why? Because the deoxyribonuclease enzyme destroyed all DNA found in the strain S, so the bacterial transformation doesn't occur. So the best answer is D. Another questions. Bacteria R will not be transferred into bacteria S. That's right. The amount of DNA in ovum of a female aphid insects that produces females by parthenogenesis is its amount in the ovum of aphid that's produced male. Pay attention, please, in this point. There is a difference between the number of chromosomal set, amount of DNA. Number of chromosomes constant in the same species. both of male and female in the human being, 46 chromosome. Chromosomal set, 2N, all somatic cells. N in gametes, both of male and female, sperm and ovules. Amount of DNA increase before the cell divisions. Normal amount of DNA 46. Before the cell divisions, the amount of DNA is duplications. By replications, the DNA it transfers from 46 to 92. So the amount of DNA increase before the cell division. And the prophase and the metaphase is precede from interphase. Okay? The amount of DNA increase to doubles. Why? This is the cells is divided mitotically. So the amount of DNA in these cells is replication the DNA. So the form of chromosome is consists of two chromatids connecting from the center with the centromeres. Normal rate in chromosome, in normal case, it consists of only one chromatide. But before the cell divisions, the chromosome, it's become two chromatids connecting from the center with the centromeres. Each chromosome contains one molecule of DNA, but in two chromatids, each chromatid contains one molecule from the DNA. So the amount of DNA, it's increased before the cell divisions. It duplications. By replications the DNA for a specific type of enzyme, inshallah, we will discuss in next questions. Okay? The amount of DNA is the ovum of the female aphid insects that producing a female by parthenogenesis. 2N. Okay? The amount of ovum of aphid insects is N. Half or double or equals or quarters. The best answer is doubles. The best answer is double. Dear students, thank you a lot for you. فاصل قصير ونعاود إن شاء الله بعد الفاصل لاستكمال الحلقة النهائية، حلقة المراجعة النهائية لمادة الأحياء باللغة الإنجليزية. إن شاء الله نتقابل بعد الفاصل. أترككم في رعاية الله وأمنه.
تالتة ثانوي هانت خلاص، أنا قد التحدي، خطوة كمان والبكرة هعدي. تالتة ثانوي، تالتة ثانوي، تالتة ثانوي هانت خلاص، أنا قد التحدي، خطوة كمان والبكرة هعدي. تالتة ثانوي، تالتة ثانوي.
أعزائي طلاب الصف الثالث الثانوي، أهلا بكم بعد الفاصل القصير لنعاود الاستكمال حلقة المراجعة النهائية لمادة الأحياء باللغة الإنجليزية. توكلنا على الله. Okay. Now we are going to focus about another questions that related to DNA, deoxyribonucleic acid. Please pay attention and follow me. What is the number of turns that are present in the piece of DNA double helix containing 1,000 nucleotides? Pay attention, please. DNA is called deoxyribonucleic acid. Deoxy, that means that deficiency of oxygen. It consists of ribose sugars or deoxyribose sugars. Deoxyribose sugars, that's right. Why? Because deficiency of the oxygens. But ribose sugars found on RNA, found in RNA. But in DNA, deoxyribose sugars, pentose sugars, it's types of the deoxyribose sugars. Pentose sugars, types deoxyribose sugars found in DNA. Okay? DNA consists of 5 carbons. Carbon number 1 leading from nitrogenous bases. Nitrogenous bases is divided to groups: purine, double ring, adenine and guanine. Pyrimidine, thymine and cytosine. Adenine plus guanine equal thymine plus cytosine in DNA double strand. Okay? The connection DNA consists of nucleotide. Nucleotide, deoxyribose sugars, carbon number 1, nitrogenous bases, carbon number 5, leading from phosphate groups. What is the binding between nucleotide? Carbon number 5 connecting with the carbon number 3 with the next nucleotide. Okay? Carbon number 5 connecting with the phosphate group, connecting with the carbon number 3 with the next nucleotide. It's called sugar phosphate backbone. Again, carbon number 5, connecting with the phosphate groups, connecting with the carbon number 3 with the next nucleotide, forming the sugar phosphate backbones along the side of the helix. Okay? One strand and another strand. At the same structures in one strand and another strand. The nitrogenous bases that's found between adenine and thymine. For example, adenine connecting with thymine to hydrogen bond. Guanine with the cytosine, 3 hydrogen bond. Okay, that's right. According to Watson and Crick, DNA, double helix, like ladders. Sugar phosphate backbones outside the ladders. Ranges of the ladder, that's 3 pairs nitrogenous bases. Adenine connecting with the thymine, 2 hydrogen bond. Guanine with the cytosine, 3 hydrogen bond. Okay, that's right. 2 strands always at the same distance from each other in the helix. Why? Because in the another strand and the nucleotide double ring, it's opposite single ring. Double ring, single ring. Single ring, double ring. So the distance between 2 strands at equals. Okay? The strand of DNA 1' from prime 5 to prime 3, prime 5 is connecting with the phosphate group. Prime 3 connecting with OH hydroxyl groups. And the opposite strand is run from prime 3 to 5. One strand 5 to 3 and another is opposite. Why the 2 strands in opposite direction? To give a proper correct between the hydrogen bond, to give a proper correct about the hydrogen bond between nitrogenous bases. To give a proper correct about the hydrogen bond that's bending between 2 nitrogenous bases. All sugar phosphate backbone covalent bond. Nitrogenous bases is connecting with the pentose sugars, deoxyribose, covalent bond. All its type of covalent bond. Another type of bond between 2 nitrogenous bases, adenine connected with thymine, 2 hydrogen bond, guanine with the cytosine, 3 hydrogen bond. That's right. DNA, it contains a ternus. Each turn, it contains 20 nucleotides. 10 another strand and 10 in opposite strand. Each ternus, it contains 20 nucleotides. Each ternus that's contain 20 nucleotides. Okay? 100 turn. تمام? What's the number of nucleotides? Okay? 110. 110. Each turn contains 20 nucleotides. 100 by 20, the number of nucleotides. Number of nucleotides divide 20 number of turns. That's right? Okay. Now we are going to focus about this questions. What is the number of turns that are present in a piece of DNA double helix containing 1,000 nucleotides? Number of turns, each turn contains 20 nucleotides. Number of turns, 1,000 divided 20 equal 50 turns. The best answer is 50 turns. That's right. Another questions. What is the number of nitrogenous bases that's from 2 hydrogen bond with the bases of the following strand in DNA molecules? Cytosine, guanine, adenine, thymine, cytosine, adenine, guanine, adenine. Cytosine connecting 3 hydrogen bond, guanine 3 hydrogen bond, cytosine 3 hydrogen bond, guanine 3 hydrogen bond. What is the number of 2 hydrogen bond? 1, 2, 3, 4, 5. Why 5? Adenine, thymine, adenine. The number of adenine and thymine in this strand is 5. Why? Because the adenine connecting with thymine 2 hydrogen bond and the guanine with the cytosine 3 hydrogen bond. The number of the nitrogenous bases forming 2 hydrogen bond is the best answer 5. That's right. Okay. Another question. What is the number of nitrogenous base pairs? Pay attention. Base pairs. That means that pairs. In a part of DNA molecules that's containing 150 turns. 150 turns. That means one strand, 150 nucleotides, another strand, 150 nucleotides. Okay? That means that each turn contains 20 nucleotides. 150 by 20 equals 3,000. That means that pairs, 1,500 pairs. 1,500 pairs. So the best answer is C, 1,500 pairs. That's right. Okay. What does each chromosome is representing in the eukaryotic? In eukaryotics, chromosome in normal case, it consists of only one chromatide. Each chromatide contains one molecule from DNA. Before the cell divisions, the chromosome consists of two chromatides connecting from the center with the centromere. Each chromatide, it contains one molecule of DNA. So the DNA is replicated before the cell division to receive a copy from original cell genetic information. That's right. That means that in eukaryotics, each chromosome contains one molecule from DNA to the best answers. What does each chromosome represent in the eukaryotics? A single DNA strand? No. One molecules of DNA is the best answer, one molecules. Two molecules? No. One molecule of DNA and one molecule of RNA? No, the best answer. RNA not found in chromosome. RNA is transcribed inside the nucleus from DNA as transferred through the nuclear pores found in nuclear membrane into cytoplasm to complete the translation process to formation the protein synthesis inside the cytoplasm. That's right. Okay? The best answer is B, one molecules from DNA. Mutations appears on the offsprings when they take place in the muscular tissue? No. Mutations, it appears. Through mutations, it transfers from generation to another generation, from the parent to the offspring. It's called true mutation. Doesn't transfer to the offspring, it's called false mutations. What is that related to the mutations? Muscular tissue? No. Scoritum cells of the testis? No. Primary germ cells. Why? Because the primary germ is forming the sperm. Sperm is found in the testis, it produces releasing from the testis to fused with the oviums, forming the new embryo. There is the disturbance it's occurred in the gametes. In the primary germ cells, the mutation is transferred into the sperm. The best answer is primary germ cells. That's right. Another questions. What happens in a case of the absence of regulatory proteins from the chromosome? Pay attention, please. In this point, please pay attention and follow me. The specific type of protein, histone proteins, non-histone proteins. Histone protein, they are well-definite group small structure proteins. It consists of only two main amino acids, arginine and lysine. Both of arginine and lysine, it contains alkyl group R's, it's a positive charge. Bending with the negative charge of the phosphate groups. Histone found only in the eukaryotics. And DNA coiled around the cluster of histone. Histone, well-definite group. Small structure proteins consists of two main amino acids, arginine and lysine. Non-histone protein, they are well-definite group structure. They are the heterozygous groups with the many function. Heterozygous groups with the many function. Regulatory and structures. Structure protein play an important role in special DNA inside the cells. Organize the DNA inside the nucleus. So that's called the specials. Site, special, that means the site inside the nucleus. Site. Regulatory, non-histone protein determine whether DNA code will be used in making proteins, enzymes, RNA. There is a difference between histone proteins and non-histone protein. Non-histone protein is related to structure, non-histone, regulatory, non-histone protein. That's right. So, what happened in a case of absence of regulatory proteins from the chromosomes? DNA structure change? No. Chromosomal shape inside the cell change? DNA parameter can be determined. The best answer, DNA parameter can be determined. When the transcriptions RNA from DNA inside the nucleus, a specific type of enzyme is called RNA polymerase enzymes. A specific type of regulatory proteins. Structures, regulatory proteins. Regulatory protein. Non-histone regulatory proteins. This enzyme binding with the DNA, forming the perimeters. After the perimeters, starting with the transcriptions mRNA, messenger RNA. By starting with start codons AUG from the nucleotide TAC following the parameters on the strand DNA that's run from prime 3 to prime 5. Okay? So the RNA is transcribed from the strand 3 to 5. Why? Because RNA polymerase enzymes run from direction 5 to 3. So the complementary strand, it is transcribed from DNA and RNA from 5 to 3. Starting with the start codons, AG, and the stop, ending with the stop codons may be A, UAA, UAG, UGA. There's the 3 types of the stop codons, UAA, UGA, UAG. Only one found on mRNA. Only one from 3 types of the stop codons. Okay? So the best answer, DNA parameter can't be determined. It is the best answer in this question. That's right. What is the first nucleotide triples that follow the parameters on DNA during mRNA transcriptions? This is answered before in the last questions. It's TAC. Pay attention, please. T, thymine. In the opposite mRNA, adenine. A, in opposite mRNA, thymine. But thymine not found in RNA, it replacing pyuracils. So the TAC is following the parameters. It is translated into TAC, AUG. It is representing the start codon on mRNA. That's right. Another questions? The best answer is TAC. In eukaryotics, RNA transcriptions occur in all the following cases, except the presence of regulatory non-histone protein in DNA. That's right. Transcriptions that occur in the presence of regulatory non-histone protein, for example, RNA polymerase enzymes. Number B, presence of DNA in the form of coiled nucleosome. No, this is the best answer is B. Transcriptions. Replication DNA doesn't occur in a coiled nucleosome, but it formed a nucleosome. Pay attention, please. There's a difference between nucleosome, chromatins. Okay, nucleosomes, DNA coiled around the cluster of DNA, DNA coiled around the cluster of a histone protein. Shortened DNA, 10 folds. But chromatins, DNA coiled around the cluster of histone proteins, shortened 100,000 times by effect of the structure non-histone protein. So the replication and the transcription that occur in the nucleus doesn't occur on DNA in a form of chromatin or coiled nucleosome, but occur in the form of only nucleosome. Coiled around the cluster of histone protein, shortened DNA 10 folds. So the best answer is the presence of DNA in the form of coiled nucleosome eukaryotics. RNA transcription occur in all the following cases except this point. That's right. Another questions? Which of the following representing nucleotide triple for a stop codon on the DNA strand? AA, this is the choice translated to found in the strand of RNA. UUU, no. AGG, UCC, no. ACC, UGG, no. ATT, UAA, the best answer is D. Why? Because the stop codon is the 3 type. Stop codon is found in the 3 types. Number 1, UAA, UGA, UAG, UAG. The best answer, ATT, is translated to UAA. Pay attention, please. DNA consists of 2 strands. One run from prime 3 to 5, while another complementary 5 to 3. 3 to 5, complementary 5 to 3. The complementary strand on DNA at the same mRNA, but replacing thymine with uracil. Anticodon on tRNA at the same template DNA that run from 3 to 5 by replacing thymine to uracil. That's right. Another questions? The best answer, ATT. The following sequence representing 2 segmenting of DNA X and Y, study them then answers. X run from 5 to 3, complementary 3 to 5. Y, 5 to 3, complementary 3 to 5. Which of the following is correct regarding this sequence? mRNA can't be transcribed from segment Y? No, can't transcribed. The segment X contains more phosphate groups? No. Segment Y needs high temperature for separating two strands. The best answer C. Why? Because segmented Y contain guanine and cytosine more than the segmented X. Both of guanine and cytosine connecting together by 3 hydrogen bonds, they need more temperatures to breaking down between them. The best answer is C. That's right. Okay? The gene whose affecting is different in all human beings and can't be cloned except from its owners is located in the chromosome number 8. That's this chromosome related to the fingerprint. Fingerprint, it is related to in this. Finally, this questions is related to, say, another type of questions. Gene X is formed from 150 pairs of nucleotide. What is the number of amino acids that's unconsistent from the resulting proteins? Minus 150 minus 3 and divide 3 give us finally the number of amino acid. Why? This is the number of nucleotide. It contained the number of stop codons. Analyze a genetic materials virus gave the following result concerning the ratio of nitrogenous bases. Guanine equal cytosine. Uracils equal adenine. Okay? What is the kind of nucleotide acid the virus has and why? It contains uracil. That means that virus is RNA. Why? Because it contains uracils replacing with the thymine. Thank you a lot for you, dear student. I wish I could see you again. إن شاء الله بالتوفيق والسلام عليكم ورحمة الله وبركاته.
تالتة ثانوي هانت خلاص، أنا قد التحدي، خطوة كمان والبكرة هعدي. تالتة ثانوي، تالتة ثانوي.